Okay, with that, let's get started and start with some of the GlyGen tools that we haven't explored in the last few weeks. So you have seen the web page in most of our talks. Hopefully you also used it yourself and explored it a little bit. We talked about the Glycan and the protein search, and now we are jumping into something we call a super search, or you can also call it a knowledge graph. So that's a additional mechanism how you can explore and search in GlyGen, and it represents the information that we have in the system in a visual way, where you can see these boxes, these edges, and a bunch of numbers. For the beginning, I will use that button here to simplify the view, which will remove some of these numbers, and that will help me hopefully a little bit to explain how that graph works. So the main concepts, as you know, are glycans and proteins in our system. And you can see on each of these boxes, there is the numbers of glycans and proteins that we have in GlyGen, the total number in the beginning. But you can also see we have these affiliated concepts like glycan have motifs, and glycans come from organism, proteins come from organism, proteins come from genes, which come from organism. Proteins may be involved in diseases. Proteins may be enzymes, and proteins have sites where glycans can be attached to. So that's essentially like a visualization of our data types and how we are related to each other. And on each of them, you can see the total numbers of enzymes or diseases. Just a note to the enzymes, this is just a general annotation of an enzyme. It doesn't mean it's a glycoenzyme. You see some of them are yellow. These are actually the boxes that you can click on, and then you can go to the result pages that you have seen in the talks 2 weeks ago from Karina and myself, and from Jeet about the site search for myself and Karina for the protein and glycans. But we said that's a search tool. Right now, we haven't really done any search. So what you can do is you can click on any of these boxes, and you can apply some filters to it, some search criteria. So for example, I'm clicking on GlyGen, and you see a bunch of fields here. And you can see for each of the boxes, there is a list of categories, how you can filter that particular box. So I clicked on the glycans, so I can filter by glycan type. So let's say we are interested in some N-glycans, okay? And then another criteria, we are interested in big glycans, so we filter by the mass, and we are saying 3,000 Dalton, and we want to have all glycans which have more than 3,000 Dalton. And then another thing is we are only interested in like fully defined glycans. And as you can see, for some of them, the search criteria are just text fields, or in this case, it's a number field. And for some of them, it's a drop-down where you can select some information. You can also see that you can build a very complicated query here where you can customize your queries with and or. So in my case, I will keep it simple, where I just say, I want to have only the glycans which are end glycans and have more than 3,000 Dalton and are fully defined. But you can also build more complicated structures with and or operation in the mix. And even if you have more than 4 queries, you can keep adding additional fields there. And you can also change the order by moving things up and down. In my case, it wouldn't make any effect because everything is and connected. But if you have a mixture of and or, it may actually have an effect where on which position a query is. So, and you can also delete query option. So once you're done defining your query, you can click on search, and it will take a second, and it will recalculate the entire graph and update the numbers with based on the query that I performed. So you may remember we had something around 40,000 glycans, and we had something around 100,000 proteins, and that numbers are now all dropped down based on that glycan query. So we have 400 glycans left. We have 21 sites and 124 glycans. Proteins. You can also see that on these around 400 glycans, there are 19 different motifs. Unfortunately, you can't click on them and find them out, so you will have to go via the glycan, which we will do for a second. So we will click on that yellow box for the glycans, and you can see the 400 glycans that have been found by our query. And you see we are all fully defined, we are all pretty big, and of course we are all N glycans. But that's kind of boring because we just found glycans based on glycan properties, which, if you remember Karina's talk, you can also do with the Glycan Advanced Search. More interesting is that if you look at this relationship, we can see that out of that 400 glycans, some of them are connected to 21 sites, which means these sites have some of these big glycans attached to them. And you can also see these sites are connected to proteins, and you see we have 124 proteins. It doesn't really make sense that we have 21 sites and 124 proteins, because, you know, sites, we should have less proteins than sites. But please keep in mind that we have these enzyme annotations where we have that relationship that glycans can be built by enzymes. And Nathan was talking about that in terms of glycotree and the sandbox last week. So, and of course, these 117 enzymes that have been involved in making that glycans, they are also protein as well. So essentially, if you look at the edge, we can see 7 of these proteins actually come from the site. So, and if we click on these 7, we will actually see these 7 proteins, which supposedly have sites where some of these glycans that we searched for are attached. So, and if I'm clicking on one of these proteins, it will load the protein page. And then we can scroll down to the glycosylation section, where we already see, okay, here there is a site where one of these big glycans, actually we have multiple of them, have been reported to be attached to that particular protein. So essentially, that's a way how you can search by one property, for example, by motifs. We could also make a search where we say, okay, we are interested in a particular motif like Lewis X. How many glycans do we have in the system, and how many of these glycans are attached to proteins, and how many proteins that do have a Lewis X motif on them based on their glycans. So you can really query across different concepts, which with all the searches you have seen so far, you can't really do using them. I should say one thing, just as a comment. If you are querying on one box, let me just go here. So that's my Glycan query again. It will not allow you to perform a query on a different box. The reason is there that it's actually very complicated to make that query work across the different concepts. And there's a lot of cycles in here, like especially between organism and glycan. So if you think about that glycan query, you can see that these 400 glycans only have 3 organism annotations. Let's say it's mouse, human, and rat. Now, but organism is also connected to protein. Just because we have mouse, human, and rat now available doesn't mean we actually want to query that edge and also get all mouse, human, and rat proteins, because we started with glycans, so we are only interested in proteins that have something to do with that glycan. So we don't want to query along that edge. But if we are starting with an organism query, where we say, okay, we want to filter everything, give me all human stuff, then of course we want to query that edge because we want to have all the human proteins. So it really depends which edge to query based on which box you start with. And if you query based on multiple boxes, then some of the edges that you said you shouldn't be query, you now have to query because of the property of the second one. So it's not a problem for us to generate numbers and the results, but our problem is actually to make it work in a way that it gives meaningful numbers for all possible queries. So for now, only one query on one box is allowed. But what you can do is, if you're more experienced and you want to do more advanced stuff, you can look at the query. It's a JSON structure, and we have can provide you with documentation for that. And then we have that debug mode version where you essentially can build your own JSON query. You can either copy that one or you can build your own one and then submit it to the system. And you can also use, you don't have to use the web interface, you can use the API directly. Robel is gonna talk about API usage in an hour or so. Okay, so that is one of the tools that you can use to explore glycans in a more advanced way. The other tool I want to show you or introduce to you is the Glycan structure dictionary. So that is some work that came out of the literature mining projects. So you may remember when I was talking about the proteins, that here in the glycosylation section, we had that text mining box, which for that particular protein is empty, but for some protein is filled. So we had a small project 2 years ago for 1 year, where we essentially took 4,000 papers and we applied literature mining or text mining strategies to extract glycosylation sites from these papers. And that's how some of the, or how the information in that tab for some of the proteins was populated. We are also doing this for more papers right now, and we are adding more and more annotations. And we are also working on improving that literature mining to not just extract glycosylation information, but also other information from the protein side, like function information, tissue, disease information, and so on. But one of the side projects that came out of that one is that while we were looking for the glycosylation sites, we obviously also needed to figure out, okay, does this paper have anything to do with glycans, and how do user describe glycans? So, and what we did there is essentially we tried to find words or phrases, you could also call it glycan names, that people in papers use to describe their glycans. So we extracted them and we built that dictionary. And in fact, we are just starting with a new project where we are expanding that dictionary using machine learning approaches. So if you are scrolling down here, you will see it's a very long list of stuff. And I'm not sure how the resolution is, but I can also zoom a little bit in. And you will find a lot of phrases or words that might be familiar, and some of you might have even used in your own paper. So 2,6 branched own monos, some blood type labels, the cores for the O-glycans, more complicated phrases like fucosylated, acylo biantennary structures. And then if we go down here, we will also find things like motifs, like Lewis X. So if you're clicking on one of these entries, you're coming to that section which is dealing with that particular phrase. So you can see that's the phrase that we found in the paper. It has an ID because we give IDs to everything. It has a GlyCan accession. I will talk about that in a minute. And you can find an example term from one of the papers where that term was found in that paper. And you find a list of other papers where we found that Glycan structure term. Plus a definition that we, in most cases, got from CABI, if it was present there. And you can also find references to other databases like glycoepitope, CABI, or glycomotifs, which deal with the same or with the structure that is described by that label. You can also find a bunch of synonyms, function description, disease annotation, Wikipedia link if there is one, and if there is that particular glycan with that name mentioned in the Essentials of Glycobiology. You may remember that's that book which is available on the NIH webpage. You can find that chapter where it was mentioned. Now, that's just a name, right? Now, what we are trying to do, and we are working with Nathan on that, is we are trying to associate that names that we found in the papers with glycans that we have in GlyGen, so that you can actually search using that name and find the corresponding glycans. So not just the motif in that case, but also all the glycans that have that motif on them. So we started with some manual creation where we found the corresponding glycans or GlyCan accessions for it. And you can see the cartoon over here, which describes that name. And Nathan can use the substructure search that he presented last week to essentially match that with the glycans. But for some of them, if you scroll down a little bit, like this one, there's a term like monosilated, right? And you can't really make a substructure there that really describes that term very well. You could make like a sialic acid and try to make a substructure search, but then you still have the problem. It's supposed to be monosilated, and the substructure search would find also, disilated or more structures with more sialic acid. So there we are using combinations of, composition search, search by glycan type, search by substructure to essentially work on that. Right now, that's still work in progress, but that's like a little, outlook in the future, so that you will be able to use these words in, for example, the GlyGen general search up here, and it will respond to you with some answers. It will give you answers already right now, where, for example, via motives or function description or other description, you will already find something. But you can imagine we have a lot of more actually monosilated structures in the database. And once we are done with that project with Nathan and we have expanded the dictionary, that will also allow you to perform that search. So if you look at that structure, we find some of them that we would agree on. And right now we also find some of them which, look strange. Why is this associated with monosilated? But, just to show that to you, I know why. If you are going down to the biomarkers, you will find that that glycan is actually involved in the biomarker, which is a ratio between the monosilated version of a glycan and that non-silated version of the glycan. And because that word was in the description of the biomarker where that glycan is associated with, it was found in our current search. But again, that will be much more meaningful, or the search will be much more meaningful and powerful once we are done integrating the glycan structure terms into the GlyGen. But in itself, that structure dictionary is already very useful and might be useful for some of you. And you can use it either here on the wiki page, or there is also a data set where you can download it as a CSV file. Okay, now we are leaving GlyGen and we are looking at some affiliated projects or some tools which are very important for GlyGen as well. That's why we want to introduce to them. You may remember pretty much everybody or most of us have talked about that SNFG, the symbol nomenclature for glycans. Mike started with it, and he also introduced that web page, which is essentially the definition of glycan cartoon representation. And if you're using glycan cartoons, please really use their definition. So use their color, use their shapes, don't invent your own stuff. That's supposed to be the standard. Please use it. And like I said, you can find all the shapes. You can find the color definition there, I think further down a little bit where you see examples, and you can also find the exact color codes that you are supposed to use. So please use it. Now the question is, how can you make your own cartoons if you are not a bioinformatician and you don't want to build your own tools? Well, there are a bunch of drawing tools available. And I will show you some of them and, just to expose you to a bunch of them. So the first one is called GlycanBuilder, and that comes out of a GlycoWorkbench project. Maybe some of you know that. GlycoWorkbench was really a very helpful mass spec tool, but it also had one of the first draws for glycan structures. Although it doesn't support the newest SNFG definition and the right colors, but it's still very powerful. And we come back to that in a minute. So this is a web-based tool which you can use to build your glycan structures. And if you're familiar with GlycoWorkbench, you already see it's very similar. So the first thing you can do is you can start with a base structure. Let's say I want to make an N-glycan, which is bisected. So I already get that template. And then I can start expanding it. I can click on the monosaccharide where I want to add something, and then I can add, for example, that GLUCNAC. And you can see the linkage right now, and the anomer is undefined. So I go over here and then I change it to a beta, and that's supposed to be a 1.4 linkage. Oops, let me disable that one. And there we have our 1.4 linkage. And that way we can really very quickly build your Glycan structures that you want to have, right? And then what you can do from this page, you don't have to take a screenshot, although it might sound tempting. You can actually export that into an SVG files if you work with vector graphics, or into a PNG file if you want to have a pixel graphic, like for a PowerPoint slide or something like that. You can also change the display. For example, if you don't want to see that mass line, or if you don't want to see the linkage information to get a more compressed version of that Glycan, you can disable it. You can rotate it if you want to have it in a different orientation. And what you also can do is you can not just make an image out of that, you can also export that into a text format that you might be using in another program. So GWS is one of the formats. That's the format GlycoWorkbench was using. I will talk about a little bit about that more in a second. But you also have a bunch of other formats, including works. And if you just click on encode, you will get the work string for that Glycan that you have just drawn. So you may remember when we are talking about Glycan, we have that GlyToucan IDs, and that you should be registering your Glycans and GlyToucan, and then use the GlyToucan IDs in your paper. So that's one way how you can do that. GlyToucan expects different sequence formats. Works is one of them. So you can use one of the drawing tools to draw the structures that you want to register, and then you can copy that sequence string into GlyToucan and either get the ID for that sequence, or register it and get, you know, the new ID for that sequence that you then can use in your paper. You can also, if you have a sequence, when you, for example, you come from another web page, you can also put a sequence in. Let me just quickly go to see up here. That's motive. That's not what I want. We are on the GLYCAM page, so you can go down to the digital sequence section. There's a bunch of digital sequences. One of them is works, so you can copy that to your clipboard. And we are going back here, dropping that here, and then we are selecting works, and we click on import. And then that structure that we just saw on the web page, you have it here, and you can, for example, further modify it. So that's a way, for example, if you're looking for a GlyToucan ID and you can't find exactly that Glycan you're looking for in GlyGen, or, you know, in any other database, you can just take that sequence in one of these drawers, or a close sequence to one of these drawers, and start modifying it. Downside with these web tools is you cannot actually save them, right? You're working on them, then you get your image or you get your sequence out of this, but the moment you leave that page, you know, it's gone. I will show you also a bunch of tools where this is not the case. There is also an offline version for that, for GlycanBuilder, which you can download on that page. You will also find that in that handout that I gave you. You don't have to worry about the compilation. If you click on that link, you come to a page where you can download a Mac version and the Windows version that you then can use on your desktop. And that's how that looks like. Again, for people that have worked with GlycoWorkbench before, they will find that pretty similar. And here it works exactly the same way that we just saw the web version. You just have a bunch of more options, what you can do. So again, we can start, in this case, let's start with an end Glycan, core 5. So it starts with that one. And then, you know, in the same way as we saw before, you can keep extending that cartoon that you are drawing here and customizing it with, you know, more detailed information if that information is available. And also here you have the option to save that into different graphical representation. You have a few more formats here, but again, SVG and PNG or JPEG might be what you're looking for. You can also export that into different sequences. But what you also can do, and that's the most important thing, if you're working, very often you will be working on multiple glycans. So you can, oops, sorry. You can actually, draw multiple different glycans in that workspace here, and then you can save that into a file. So in this case, it's called a GWS file, which is the format which GlycoWorkbench was using in the past. And we're keeping that alive because it has the advantage that the structures that you have drawn here, you can actually bring back to your GlycoWorkbench tool in case you're using that, for example, for MS profiling, or you can put that into Grid's Toolbox. I talk about that in a second. So you can even exchange them between different tools. And most importantly, you can actually reopen it here and continue working on that. Now, if you are working on glycomics and you have glycans that you want to share with GlyGen, and you already have that GWS format, you're actually halfway there because we have tools that can do, the GlyToken registration and GlyToken and providing you with the GlyToken IDs for the structures that you have drawn there. Okay. I want to show one more web tool. Nathan touched on that last week already because that's GlycoGlyph. Again, it's in the handout that I gave you. It's one more way how you can use a web application to drawing it. So if you, that like a workbench thing, you actually need to install something on your local system. The same will happen with Grid's Toolbox, which I'm showing you next. If you cannot install or you don't want to install, you can use these web tools. And that's one other option. So here again, you have these templates. We take the kind of BIOS. And then if we want to extend the structure, we have to say, okay, what is the monosaccharide we want to use to extend? So it's a mannose. And then that's supposed to beta 1,4 linkage. We add the linkage. And then we'll just click on that and it will add that mannose with a beta 1,4 linkage to the structure. And just to, remind you, as I said, that was on the Glycan search. If you make a structure search or substructure search, you can drop your own sequence if you have it, for example, in GlycoCT or Vox. Or if you don't have it, click on that draw and you get these window that we just saw. And then in different from Nathan, you don't actually need to copy that sequence. That's one option. But you can also just click that copy Glycan to search button and it will copy the sequence for you right in there. And then you can perform your search. Again, web tool. Oh, before I forget it, here is that little camera icon, which allows you to save that into an image. You can also here again customize the angle. So I'm actually more like a left-right person. So you can also change that and then generate the image based on that. And the last drawing tool, sorry, Zoom gets in the way. The last drawing tool that I want to show you is Grid's Toolbox. You can download it from this website. I should say that's one of the tools that we have developed. Again, you download it to your local system, you install it, and then when you start the program, you get a window like this. It's actually mostly made for mass spec, but it also has a drawing tool. So you can go to Tools, GlycanBuilder, open GlycanBuilder. And then you see it looks very similar to all the other tools. We have the templates up here. This time, let's pick a milk sugar, put it over there. You can also add multiple glycan structures. You have a very extensive list of monosaccharides, not just the Mamiya stuff, but also a lot of other of them. You can also add subservient modifications. And I will say most of that is also available in the tools I showed before. So we are all very similar, powerful. And in difference to the other tools, you can change the orientation. You can change the zoom, the size of them. You can also have this compact version where you don't see the linkage information. You have a more compressed version. And of course, you can save it into files or images. Or if you're using grids also for glycomics mass spec data, you can also build your own glycan structure database here and save it into a grids database and use that database then to annotate your mass spec spectra. Most important thing is, if you want to have a cartoon, there are a bunch of tools available that can help you doing it. So if you're still using your PowerPoint templates or your Illustrator templates or whatever is you're using, well, maybe consider switching to these tools. And most importantly, always remember to save these structures into that GlycoWorkbench files, because many groups have tools that can work with that files and do something with it. Like I said, if you want to import glycans or share glycans with GlyGen, that GlycoWorkbench file is a very good start. Okay, do you have any questions so far to the drawing tools or something like this? I will give you all like 3 or 4 minutes to explore them, at least the web tools, so you can play around a little bit with them, and also give you the time to think a little bit about questions. And if you don't have questions in 2 minutes, then we will continue with the next. So Tyler was asking if there is a link to these tools in a central place, such as GlyGen. Yes, not all of them, because like GlycanBuilder, for example, is fairly new. So let me go back to the browser. So on GlyGen, the place that you want to go is here, tools and other resources. There is a link list with other databases, but also with tools. Unfortunately, like I said, not all of them are there. If you're looking specifically for the drawing tools and you're not happy with the list that I provided you in that handout, you can also go to the SNFG webpage, and we have here some of these drawing tools also mentioned. So you can find GlycanBuilder here, and there are also other tools, but again, it's not a complete list. But honestly, try the tools that I gave you. If for whatever reason you're not happy, send me an email. We can see if we find another one. There was a template. Okay, there was a question if there are templates for plant oligosaccharides or polysaccharide structures. So no, none of them has templates for plant or bacterial polysaccharide structures, but that just means there are no templates, right? You will find all the monosaccharides that you need in here. Like, for example, you need arabinose and ribose and GLUC-A, and you just can start building them yourself, right? And there's also the option for making polysaccharides. So we do it like that, where it adds the bracket here, and you can add, the repeat information. And in most of these tools, I may, give me 1 second. I think GlycoGlyph actually doesn't do, yeah, it doesn't do the polysaccharides, because it's really meant as a, input tool for querying databases right now. But most of these drawing tools, especially GlycanBuilder and grids, we can make these structures, right? But yes, you don't have templates. But what you also can do is, if you have like a common motive for your plant structure, you can just draw that the first time. And then what you can do is, you can just copy it and then paste it multiple times, and then make the different variation of that structure that you want to make. I hope that helps. Okay. Any other questions? Okay. Well, if something comes to mind, drop it in the chat and we can talk about that. So now we are leaving GlyGen and talking a bit about affiliated projects. So one thing you at least should hear about is the GlySpace Alliance. I think Rasha and Mike both mentioned that. So that's essentially an kind of a consortium between GlyGen, the Swiss Bioinformatics Institute with Frederick Lisacek, and the GlyCosmos from Kyoko. Maybe you heard Kyoko's talk 2 days ago in the GlyGen webinar. And it's essentially an alliance between these 3 resources, where we are trying to, make sure we are exchanging data with each other, correctly exchanging data with each other, and also building no duplication, but building on top of each other new resources. So if you're going to the GlyGen webpage, for example, you can really see that active, because if you're, so this is again a Glycan here. And if we are going down to the protein section, you can see that that glycan is reported on that protein, and the information comes, for example, from GlyConnect. And you may also remember in the species section, we also find GlyCosmos, where the species annotation was also provided from GlyCosmos. So when we are building GlyGen in each release, we are getting information from all these databases and integrating that into GlyGen. But it's not just in GlyGen. If we are going to GlyConnect for that structure, you will also see we also have links back. So GlyConnect doesn't actually take data from GlyGen or GlyCosmos because we have a curated database system, but we are at least placing links. We are placing links to us. And then if we go, Zoom is really, if we go to GlyCosmos, we find similar information, same glycan, obviously. And you find in that section also the connections to GlyGen and GlyConnect. But GlyCosmos, for example, is also pulling information from GlyGen, for example, the species information and other information. So it's really an environment for actively sharing and also, making sure that we are using standards when we are exchanging data. Okay. Yes. So that's something just to know about. So we are really working on making sure that even if you are not using GlyGen, but you're using GlyCosmos or GlyConnect, there's always a way from these pages to GlyGen and additional information. And the same in the opposite direction. If you are having GlyGen and you're looking at, for example, glycosylated protein information, you will have a link back to GlyConnect or GlyCosmos to find supplementary information. Another initiative that I would like to mention is the Glycoinformatics Consortium. That's essentially a group where we try to get together the people working in bioinformatics in the field of glycobiology. And it's essentially it's a mailing list where we are trying to be more active now with seminars and things like that. If you want to subscribe to that mailing list, just send an email to that list and you will be added. But there's also that web page, and on that web page as well, you can find a list of databases and software tools which have been curated by the developers themselves. And we are right now updating that list to get the newest information, also start to add new tools to that list. So even if you don't want to subscribe, you may be finding a bunch of tools or databases on that website that might be useful for you. Another initiative I want to talk about is CFDE, the Common Funds data ecosystem. So some of you might be available with the Common Funds. That's a funding mechanism where NIH puts some money into a focused project to advance a certain field or a certain domain. And a few years back, one of the focus was glycobiology. There was a lot of money going into synthesizing of glycans or developing new mechanism for synthesizing glycans, glycan arrays, new glycan array technologies. And there was also an bioinformatics project where the GlyGen project came out. But then NIH also noticed, okay, we have all these focused projects and all of them have bioinformatics in one way or another. There's lots of databases generated and all of them are kind of isolated. So we are adopted within their own community where we are used heavily and people deposit data or brief data, but we are not actually connected to all these other Common Funds projects. And that's why NIH started that Common Funds ecosystem to essentially make sure that all these resources that have been created are actually connected with each other. So if you scroll down on that web page a little bit, you can see kind of a little bit outdated representation of that, where you see some of these resources and initiatives that have been created with the Common Fund money and that the ecosystem tries to connect. So you find GlycoScience, that's essentially GlyGen, but you also have other resources which focus on for example, the molecular effect of neurocycling, or we have Kids First, which has a lot of gene and RNA expression data from kids that have certain diseases, or GTEx, which is also focusing on genomics information. But there's also things like Metabolomics Workbench, which was created to essentially store experimental information about metabolomics, small molecules. And obviously the Bridge to AI project. So there's a lot of different projects with different foci, and each of them has lots of data. And one of the resources that came out of the Commons Funds data ecosystem is their data portal. And what you can see here is just, graphical representation of a chart. There's tons of alignment data, index data, sequencing data that has been submitted to the data portal. I should mention, it's not the actual data. So if you think about GlyGen, we have a lot of glycan and protein information data. It's not the actual data that we are submitting to them, like all the glycan details, all the protein details. The CFDE portal stores the metadata. So which means it says, okay, there's some files that we have in our system which talk about certain glycans or a certain disease or a certain tissue. And what you can do is in this portal, you can search for this based on this information across all these different projects. So for example, let's just go to anatomy, and you can see there's a lots of stuff. And let's say we are interested in liver. Okay, so we have different liver-associated tissues here, and each of them has data associated with them. So if you click on that little view icon here, we can see, okay, that's what we started with. That's the liver stuff. There's a bunch of collections, meaning there's a bunch of information associated with liver for certain proteins that came from GlyGen. So where we know that that protein is highly, or that glycan is highly expressed in liver. But if we are scrolling down, you can also find certain biosamples. So there's some of them from HubMap project, which is the Human Microbiome project. And then there is also information biosamples from links. And if we click on these biosamples, we will see, okay, we came from a certain subject, and there are certain files. In this case, it's a gene expression profile table that you can retrieve for them. So essentially, you can use that portal to find data files associated with, whatever you are interested in. Like I said, you can start with anatomy. You can also start with disease. So you can say, I'm interested in certain types of cancer, and then explore which of the resources have, for example, gene expression information for these cancers, or, glycan information for that type of disease that you're interested in. You can also start with proteins or glycans. In this case, you go to the data browser, you go to the vocabulary, and then you have proteins here. And let's just say I'm interested in EGFR. That's the gene name for a certain protein. And if we click on that, we will get the information about that protein. Right now, there's only information from GlyGen here. But what you also can see is there's this little section here where the CFDE portal actually pulls information from GlyGen and displays it on their web page. So it's like a summary of that protein in GlyGen. You have the links to GlyGen and UniProt, but you also can see that there are 18 glycosylation sites with 150 N-glycans and 1 O-glycan reported on that sites. And if you're interested in that particular information, you can click on that link and go back to GlyGen. And there's a similar page for glycans as well. I will, in favor of time, a little bit jump over that right now. Because the last thing I want to talk about in the 9 minutes I have left is 3D structures. So if You might remember when Karina gave her talk and she scrolled down here a little bit on these Glycan detail pages, you saw that 3D structures of that Glycan, where you see, you know, the ball and stick model, and then the SNFG 3D shapes rendered on top of that. And there is a way how you can save that PDB file that is behind that onto your local system. But what you also can see is we use a tool called GlyCam to generate glycans. Okay, so let's go to the GlyCam tool. If you're going to the GlyCam webpage, again, it's a link in that handout that I provided for you. And let's say you want to have the 3D structure of your glycan. You cannot find it in GlyGen, so you have to make a new 3D structure. So what you can do is you click on that, what are the shapes of my glycan? And it gives you different input options. You can input via text, you can input gags or polysaccharides. We also have a Glycan library, but I will start with a GlycanBuilder. And they have their own version of a GlycanBuilder. It's not as visual as the other. Let me, for some reason, okay. You can see the cartoons, but it doesn't actually build you a graphical representation in the end. But let's say we want to build something like an end-like core structure. So we have a GLUCNUG that we are starting with, and then we need a setting GLUCNUG. You can see it wants to have the linkage information from us. So that's a beta 1.4 linkage. And then we have a Manos, and that's a beta also 1.4 linkage. And you need another Manos, which is an alpha 1.3. And actually we need a branch. So we add a branch at this Manos. It's another Manos and a alpha 1.6 linkage. Okay, so, and again, I can keep building it. You can see there's a sequence down here, which is built while you're building your Glycan. Unfortunately, no visual representation, but that's fine enough. And then once I'm done with it, I can click on done. And it will actually build that Glycan structure for us. So you can already see there is the PDB file here rendered out. And you can see there is the torsion angles between these two monosaccharides. There is two favored versions. So I will just disable that and say generate only one version of that. And you see up here in the status, it's building it. And then you see a PDB link here. So now you can download that PDB file to your local system and use it for modeling or, you know, whatever you want to use it for. Any questions so far? Okay, well, while you're thinking, I can also show you a little bit more complicated than that. So let's say building a glycan is fine, right? And but what happens if you want to build a glycoprotein? Meaning you have a protein, you have a bunch of glycans attached to it. So you can also use GlyCam for that. And there's this, how does a glycosylation affect my protein option? You can, depending on how big your protein is, how many glycans you put on there, and if you're minimizing, do minimization here, energy minimization, it may take a while. So it might be worthwhile actually to put down your email address to, you know, it will send you an email once it's done. But you can also wait on your browser. So if we want to make a glycosylated protein, the first thing is we need a 3D shape of the protein. So we can go to PDB, and I just picked that protein. I already downloaded it. And here in the structure viewer, we can actually see that this is really a glycosylated protein. But what you can see, it only has two GLUCNUGs, and this is not the O GLUCNUG. This is actually N glycosylation. But very often, if you look in PDB, what happens if the glycans are truncated? So we essentially just put down the first GLUCNUG, and everything else, because it's so flexible, they couldn't resolve or didn't want to resolve, and it gets just ignored. Now, let's say we want to have that protein with some proper end ligands on there. So like I said, I already downloaded that file. So I will go to my downloads folder. there it is. I will load that. I'm clicking on continue, and then it will take a second to upload the file, and then it will analyze the file with some statistics. I'm not really interested in that part. I want to add glycans to it. And we have different options here. We can download the PDB file, which is now the PDB file with everything glyco removed. But we can also add our glycan structures to it. So for example, we can use the interactive GlycanBuilder. And that's actually the older version. Well, what I will just do is I will go to GlyGen. And you may remember that's my favorite glycan on GlyGen. And if we go down to the digital sequence section, we find multiple sequences. We talked about that today already. But for some of them, for the fully defined glycans, we also have that GlyCAM IUPAC sequence, which is exactly the same sequence that we were building with the GlycanBuilder just a moment ago. I will just copy that. Then I will drop that here. Ooh. Okay. That's okay. For some reason, it's not quite working as it was supposed to be. Sorry about that. Then I will just have to pick a glycan from their library. I take that. So that's the glycan I want to add. I'm saying I want to add that glycan to a glycosylation site. Now it analyzed the protein. It found the 2 end glycosylation sites based on the consensus motif. And I found a bunch of possible glycosylation sites. Now I just add that glycan that I selected on both sides. You can also do it individually and add different glycans on individual sites. And then if I'm clicking on download the current structure, you can, or actually I will do it without minimization because otherwise it will take a while. And that will also take a few minutes to finish. But what you will get on that web page is actually, again, a 3D representation of that glycoprotein now that we generated. And you will get on the left side a download option where you can download the PDB file that that is generated with the glycans and the protein, or other way around, the protein with the glycan attached to it. So here we see that structure is a little bit hard to see. Let me just say, select the protein, and then for the style, I want to do to the cartoon. So we see here's the protein cartoon, and here are these 2 glycans that we just added to that protein. So that's a way how you can generate, and then here are the download option, how you can generate a PDB file of a glycosylated protein, even though in PDB, that protein didn't actually have these glycans, or maybe not exactly the glycans you want to have on them.